Dear student, today we will discuss different dimensions to study DNA. So there are several aspects to gain the proper understanding of DNA. So let's talk more about DNA. Now, on the basis of the nucleotide pattern, the DNA is divided into five basic categories: bDNA, aDNA, cDNA, ddNA, and zDNA. Now, bDNA is the most stable and most common DNA, which is proposed by Watson and Crick, and it is a right-handed helix, and it carries 10 base pair per turn of the helix. So if you compare this with the A and other DNA, B, A, C, D all are right-handed, and only the zDNA is left-handed. And if you compare the base pair per turn of the helix, in case of A it is 11 base pair, in case of C it is 9.3 base pair, in case of D it is 8 base pair, and it is 12 base pair per turn in case of zDNA. bDNA can be converted to other forms depending on the humidity and salt concentration. So these are different forms of DNA that exist in nature. Now, on the basis of the structure, the DNA is divided into two, you can say, categories: linear DNA and circular DNA. So, linear DNA is the one where both the ends are free, such as eukaryotic DNA, and circular is ring shaped, such as prokaryotic DNA or mitochondrial DNA. On the basis of location, the DNA is divided into two categories: chromosomal and cytoplasmic. The chromosomal DNA occurs in nucleus and it is large and linear, whereas cytoplasmic DNA occurs in mitochondria and chloroplast of eukaryotic cell, and they are small size and circular. So this is how it occurs. Now, on the basis of number of repeats, or you can say the repeats, the DNA is divided as repetitive and palindromic. Again, repetitive DNA we have already discussed in detail. They are of different types: microsatellite, macrosatellite, mini satellite DNA, and they consist of direct repeats, maybe in tandem order, whereas the palindrome consists of invert repeat sequences, and specifically they are the site for restriction endonuclease. Now, on the basis of the expression, whether the DNA is expressed or not, it is divided into two categories: coding and non-coding. Coding DNA, the one which codes for the protein, whereas non-coding does not code for anything. And coding, the amount is less in genome. If you see the total size of genome, the coding is much more less as compared to the non-coding. And the non-coding is also known as junk DNA, since it does not code for any functional protein. Now, on the basis of number of strand, it is divided into two categories: double-stranded and single-stranded. Double-stranded almost occur in all the organism. It is more stable structure, whereas the single-stranded this type of DNA is present as genetic material in certain viruses, for example, bacteriophage 174. Okay. Double-stranded DNA can be converted into single-stranded DNA in vitro, by using heat treatment or altering the pH, or by using of certain chemicals. And this process of conversion of double-stranded DNA to single-stranded DNA is called denaturation. See, when the heat is used to denature, we call this process as melting. So um, commonly it is also known as melting when the denaturation is done by heat. Now, what is complementary DNA, which is also known as cDNA? The cDNA is the DNA sequence complementary to its mRNA and is synthesized on mRNA by use of the enzyme reverse transcriptase. So you have the mRNA, and from the mRNA we synthesize the DNA. It is done artificially by use of the enzyme reverse transcriptase, whereas it occurs in certain viruses where the virus genetic material is RNA. And This reverse transcriptase is RNA dependent DNA polymerase, and cDNA is useful. We create cDNA libraries. It is usually in cloning. It is also used as probe. So there are various uses of cDNA. Okay. Now, what are transposons? We'll discuss the transposons, which are known as jumping genes or transposable elements. Now, they are the DNA sequences that form mobile genetic elements and move from one location to new location in genome. They are part of known coding sequence. They are discovered by Barbara McClintock in 1965 and especially occurs in the eukaryotic genome. So there are two different categories of transposon based on how they move from one location to another. So let's consider the first one. Suppose this is a transposon located in a genome. This is its location 1. Now what will happen? This transposon will form RNA, okay, and from the RNA it forms the DNA. Now this is reverse transcription which is taking place. So the enzyme reverse transcriptase. Now this reverse transcriptase is actually synthesized from the sequence which are present in the transposon region. Okay, so the DNA which is synthesized is now inserted in the new location in the genome. So it will change its location from one to another. Second is different procedure. Now you can look at this transposon, and this transposon is flanked by certain sequences. First, on either side there are inverted repeats, and then again inverted repeats on both the side are flanked by direct repeat. Okay, so transposon over here have inverted repeat and direct repeat flanked sequences. So here the transposon code for the enzyme transposase. Okay, and what it do, it cleaves the DNA. Where it cleaves, you can just see the transposon sequences, the cleaving sites. Okay, it cleaves this part, leaving behind the flanking sequences, okay, the direct and the inverted repeats. Now this part is excised from DNA and is reinserted into the new location, and all these activities are done by transposases, which is again coded by transposon region. So this is how they move from one position to another, and specifically these transpositions can be used to insert foreign gene into the genome in rDNA technology. Now we will see what are sequence tag sites. Okay, they are known as STS and express sequence tags, ESTS. Now sequence tag site is a short DNA sequence with single occurrence in the genome whose base pair sequence and location is known. Okay, so they are flanked by the consensus or conserved sequences that are unique to each STS. So suppose this is the sequence of DNA which are called sequence tag sites, and they are flanked by some sequences, conserved sequences, and these sequences are different for different STS. Okay, so if the STS sequence does not vary, but the vary or not vary depends from person to person, but the conserved sequence they vary. Okay, now how they are useful? These are used by the primer to bind and amplify the STS sequence. Okay, so once we know what are the sequence of primer, then the primers are used against this, and this region could be amplified in the genome, and therefore it is possible to amplify STS in presence of other genomic DNA sequence, and they are like landmark on the physical map and genetic map of the individual. So once we know where is STS situated in the genome, with respect to that we can find out other sequences. Okay, and it's easy to sequence the nearby region of the uh, genome in near the STS. Okay, and specifically they are basically utilized during the Human Genome Project. So EST and STS derived from EST, that is express sequence tags, are the sequence tag sites which are derived from complementary DNA. So let's understand, from complementary DNA, the random clones are isolated. Now there is cDNA library, you have to understand in a way, and from the cDNA library you randomly select certain, you can say, inserts. Okay, so from the clones you will just select them, and then we will do the sequencing of these cDNA. Okay, now once the sequencing is done, you can use them as a primer and you can amplify the nearby, you can say, genes. Okay, so in this way, in EST, the cDNA sequence is used to identify the full length of the gene. Okay, and EST serves as landmark for mapping. EST provide information on gene expression, function, and evolution. So once you sequence the genes, since it only carry the coding part, okay, so you can derive lot of information. Once you know the sequence, you can derive lot of information about a particular gene. So this is how they are useful. STS and EST were utilized for assembling Human Genome Project. So once you cut the whole genome with the restriction endonuclease, different parts, then again to assemble it and to trace their location and to do the genetic mapping or physical mapping, you need to have certain sequence which could be utilized as a landmark. So EST and STS were those kind of sequences. Now, there is another term called sequence annotation. It is the process of marking. Marking means either highlighting, underlining, circling specific region of DNA, RNA, or protein sequence, along with providing the descriptive information about their structure and function. So, if you know a particular DNA sequence, then you mark those areas that this particular region is coding for this particular region is coding for this. For example, here is one DNA sequence, and here, see, I have highlighted or marked the areas, like this is a non-coding region, this is regulatory region, this is coding sequence, and it codes for what purpose. So all the functional description information it provides. Okay, now genome annotation or DNA annotation is also a related term. Now the process of marking or identifying functional element along the sequence of genome. So if we are doing it for the whole sequence, then we say it is genome annotation, and this is important because sequencing genome without knowing its function is useless. Even though we know the sequence and we don't know which are the coding region, which is what, then what we are going to do with this. So it is very important that we must tag this particular region is important for this particular function. So the genome is marked with the regulatory genes, structural genes, coding, non-coding region, etc. So these were few little extensions of DNA, you can say, which will help you to understand the DNA in a better way. Thank you.